Welcome to a brief overview of electricity in the nervous system. Before I begin this presentation, I want to remind you all that the brain is just meat with electricity in it. The study of electricity in the nervous system dates back to the 1700s. Luigi Galvani is recognized as the man who discovered electricity in animals. This serendipitous discovery occurred when Galvani's assistant touched an exposed sciatic nerve in the frog with a metal scalpel that had picked up a charge, which resulted in contraction of the frog's leg. The sciatic nerve is the longest nerve in the human body, which runs from the base of the spinal cord to the big toe of each foot. These neurons can exceed 1 meter in humans. Here's an overview of a neuron. The dendrites are the branch-like projections from the cell body that receive signals from other neurons. The cell body, or the soma, is the part of the neuron that contains the nucleus, as you can see here. So this is the part of the neuron that carries the genetic information. The action potential, or electric signal, is generated at the beginning of the axon, called the axon hillock, and this action potential is propagated down along the axon until it reaches the axon terminals, which then pass on this electrochemical signal to the next neuron. It is now well understood that neurons propagate signals through an electrochemical gradient. The electrical part of it is the charge difference across the membrane, and the chemical gradient is the concentration of ions on either side of the membrane. It wasn't until the last 20 to 50 years that the chemical gradient was actually established. Now we can think of a neurocell, as having a 7 nanometer thick cell membrane and various concentrations of sodium and potassium ions on either side of the membrane. Now this gets more complicated. Calcium actually plays an integral role in conduction of neurotransmission, but we'll focus on sodium and potassium ions right now to simplify things. To maintain the resting membrane potential of a negative 70 millivolts, we have sodium-potassium pumps, which pump 3 sodium ions out for every 2 potassium ions they pump in. You can see that there are also sodium and potassium channels which allow these specific ions to flow down their electrochemical gradient when a nerve cell fires. The pumping of sodium and potassium ions creates an electrical potential difference, much like a battery, which generates an electromotive force. As mentioned before, the inside of a neuron has a resting membrane potential at negative 70 millivolts. We say that the membrane is polarized because there's a potential difference across the membrane, or a membrane potential. This is the state of neurons when they're not firing. This is definitely an oversimplification. The inside of the cell is not always negative 70, as it is constantly fluctuating. Here's another model of a potential difference between 2 plates, shown on the left, and a simplified view of a neuron with a positive potential on the outside compared to the negative potential on the inside of the neuron. Here's a quick example problem. In this example, we are told that the thickness of a nerve cell typically is, the cell membrane is typically 7 nanometers, and we know that the potential difference across the membrane is negative 70 millivolts. So, plugging this into our equation of the electrical potential equals delta V over D, we find that the electrical field is 1 times 10 to the 7 volts per meter. Since the cell membrane is not a good conductor, we view these charges as being inside or outside the membrane. This makes the cell look like a capacitor, with 2 charge conductors separated by an insulator. The cell membrane has a resistance because it resists charges from the ions moving across it. We model the cell membrane as an RC circuit because it has a resistance and a capacitance. For this reason, the membrane will have a time constant. Shown again on the left is a neuron, and on the right we can see how neurons communicate. So the presynaptic terminal is the dendrite, the postsynaptic terminal, is the dendrite of the next neuron, and the presynaptic terminal we can think of the axon terminal. Surrounded by the pre- and postsynaptic neurons are astrocytes, which are a super cool star-shaped glial cell or neuronal support cell that limit the diffusion of neurotransmitters across the synapse, and they play crucial roles in regulating neurotransmission. Neurons fire because their membrane potential changes when neurotransmitters from one neuron's presynaptic terminal go across the synapse, or the space between neurons, and bind to receptors on another neuron's dendrites. This firing of a neuron is called an action potential. In more detail, when neurotransmitters bind to the postsynaptic terminal, changes in the membrane occur in the proteins, and sodium ions flow into the cell. And since sodium ions have a positive charge, they depolarize the membrane to about 40 millivolts, positive 40 millivolts. When the membrane gets to positive 40 millivolts, potassium channels now allow the efflux, or exiting of potassium channels. We know that the inside of a cell can become more negative if we bring negative charges in or send positive charges out. So to bring us back to a negative 70 membrane potential, we push potassium ions out. This is such a robust response that we actually bring the membrane potential to negative 80 millivolts, which is lower than the resting membrane potential. This is called hyperpolarization because we are more polarized than we are at a resting membrane potential. After the hyperpolarization, the cell can be brought back to its resting potential at negative 70 millivolts, and the cell is now ready to be depolarized again. After the cell fires, there is a brief resting or refractory period during which the cell cannot fire again. It's fascinating because these potential changes occur in milliseconds, which allow neurons to fire about 200 times per second. This same process occurs with muscles at neuromuscular junctions. So, looking at the nervous system as a whole, signals are transmitted through the body in the same way that we mentioned through action potentials in neurons, and motor neurons are the cells responsible for sending signals to muscles. And as previously mentioned, these axons can be 1 meter long in the sciatic nerves. In the neuron, the action potential is generated at the beginning of the axon and travels down the axon in a wave of depolarization. This wave of depolarization occurs because depolarization resulting from the opening of sodium channels at one region of the axon causes depolarization of neighboring areas of the axon. This depolarization looks like a wave that travels along the axon towards the axon terminals. In the blue here, we can see that immediately after the depolarization, in yellow, the axon is repolarizing because of the efflux of potassium ions. So this part of the neuron is in the refractory period where it can't fire again. Now, to increase the speed of neurotransmission, the axons of neurons are insulated with myelin, which is a fat substance that wraps around the axon again and again. Schwann cells are a type of glial cell or neuronal support cell that is responsible for wrapping the axon with myelin in the peripheral nervous system. Oligodendrocytes do this in the central nervous system. There are spaces along the axon that are not wrapped with myelin, called the nodes of Ranvier, which have a high concentration of ion channels so that the depolarization events happen at these nodes and essentially jump over the myelin sheath to the next node in a process called saltatory conduction. And this process significantly increases the speed of neurotransmission. Here are the nodes of Ranvier, which you can see between the gaps in the myelin sheath. And this is where the potential differences are generated. The fluid inside the axon acts like a resistor regulating current flow, and the myelin sheath acts like a dielectric of a capacitor, insulating the space between the plates of the capacitor, or in this case, the neuronal membrane. Okay, but how exactly is the speed increased by the insulation of myelin? Well, the switch is closed and the action potential is triggered at the node through influx of sodium. This then drives a current down the axon and charges the capacitance of the next region of the membrane. Once this next region reaches a certain threshold, around negative 55 millivolts, the action potential is transmitted to this region, and sodium comes in and current is transmitted further. This process continues until the signal reaches the axon terminal, where it's transmitted to the next neuron. Now at this point, you have to be wondering how fast this action potential moves down a myelinated axon. And lucky for you, we can calculate that. We can call the critical time for propagation the time constant, and the resistance between nodes is 25 mega ohms, so 25 times 10 to the 6 ohms. And the capacitance of the membrane is 1.6 picofarads. With this information, we can use the equation time constant, characterized by the Greek symbol for tau, equals R, axon, resistance of the axon, times C, membrane, capacitance of the membrane. When we plug our numbers in, we get 40 microseconds, and we can plug this answer into the next equation, velocity equals length of the node over our time constant, again, Greek symbol for tau, And we are given that the nodes of Ranvier are 1 millimeter apart, so we can plug these numbers in, and we get 25 meters per second, which translates to approximately 56 miles per hour. I hope you enjoyed this overview of neurotransmission and now understand that neuroscientists are really just physicists who focus on the nervous system, and maybe a little biochemistry in there too. But really what it comes down to is physics. Now, before you leave, I want to remind you that the brain is the most important organ you have, according to the brain. And you think with your brain, not your heart. And here is just a little image that hopefully makes you laugh. Thank you for attending this presentation, and have a nice day.